All right. Welcome, everyone. Today is the last of the fall seminar series for applications of engineering in medicine and biology, and I'm very pleased to introduce you our today's speaker, Dr. Berk Usta, who's an assistant professor at Harvard Medical School and Massachusetts General Hospital, and he's part of the Center for Engineering in Medicine and Surgery, the same department that I belong to. So Dr. Usta is an old friend and colleague whose research interests are wide range, starting from complex fluids to tissue engineering and biopreservation and application of micro devices to biological problems. He received his bachelor's degree in chemical engineering from Boğaziçi University and PhD again in chemical engineering from University of Florida in Gainesville. He later joined Center for Engineering in Medicine as a postdoc and stayed on and continued his career here. So Dr. Usta has been part of big projects during his career, including the NIH DARPA initiatives of tissues and humans on a chip. He's also part of the Supercooling preservation team, where his group leads the efforts on preservation at the cellular and tissue level, as well as the use of microfabricated platforms and computational models to better illustrate the preservation outcomes. His work is funded by the NIH and Shriners Hospitals for Children, among others. He also serves as the associate editor of the Technology Journal. So with that, I'd like to leave the floor to Berk. And we're happy to have you.
Alrighty, thank you, Başak abla. I'll put on my slides and we'll go from there if I can find them. Alrighty, let's see. Okay, I'm hoping everybody can see my slides. Başak abla, thank you, and all the other organizers of this seminar series. It's really exciting to be here. And Başak abla is being very humble when he says friend and colleague. She actually happens to be one of the most important mentors in my experimental work. I learned pretty much everything from her in the lab. So with that, I'm gonna try to cover a lot of space today. It's really wide-ranging topics, and I think it fits the theme of the seminar series really well. We're going to look at many applications of engineering in medicine and biology. So I'm going to try to spend enough time on each one of them as much as possible. With that, I just want to remember a good leader that we had, whose ideas still live on with us today, a leader that believed in science, and hopefully today marks a milestone that we have leaders again that believe in science and trust in it. With that, I'm going to just give you a brief overview of the research that I conduct. My focus at the Center for Engineering Medicine and Surgery now is creating bioengineered solutions. These could be tissue models or other kind of cellular therapies and their dissemination thereof. The dream here is to create tissues and therapies, but then provide it to end users in an off-the-shelf manner. Throughout the years, we've done a lot of work in these. The blue ones are the two big trusts that I work on, which is engineered tissues for health and disease, and the other is the biopreservation of cells and tissues. In the engineered tissues area, we were one of the very first groups to create a 4-cell model of the liver in a microfluidic model, which lived up to 4 weeks. This was part of the NIH and DARPA initiative that Başak abla mentioned, we also work on 3D models of the liver, as well as high fidelity, high throughput models. I'm going to talk more about these today. In the biopreservation area, I actually got started on this when I arrived at the center. This was through the leadership of the other Uygun in the lab, which is Dr. Korkut Uygun, who started the supercooling efforts. Then I actually started with the cellular preservation with supercooling. But then we pushed the envelope a little bit with deep supercooling, which I'm going to mention today. Another important area which supports these two main thrusts is biomaterials. I'm going to try to give you a flavor of that, but basically we developed models to support the tissue as well as materials which reduce the problems in microfluidic tissue engineering. A more recent area of interest for us is to look at circulating biomarkers in tissue toxicity, where we can look at drug toxicity, but also their signatures in disease. Specifically here, we're targeting exosomes and what kind of signatures they provide for toxicity and disease. I'm not going to talk about these today because they're pretty new. But so this is the outline that I'm going to go over today. I always like starting with the question why, like why we do what we do. And then I'm going to tell you about why there's renewed interest in microfluidic tissues. And then I'm going to give you a flavor of the work we do on microliver tissues. I'm going to primarily focus on this chip we call metabolic patterning on a chip. It's pictured on the right here. And I'm going to tell you about how we capture the heterogeneity of the liver in normal physiology, but also its heterogeneity in disease. These are still in its infancy, but we're making progress here. Then I'm going to tell you a little bit about the biomaterials work, mostly the work of one postdoc. Hopefully she will join us too. This is about solving problems of PDMS for its use in critical applications and how we develop smart copolymers for it. Then I'm going to switch gears and tell you about why we preserve things and why supercooling is a good alternative. And then I'll tell you a bit about what deep supercooling is and how we apply that. So with that, I'm going to start with the microengineered tissues. And I'll first tell you why. As I told you, I like this question a lot. And I think the most important why here, or the motivation for us to develop in vitro technologies, is that we want to know the safety of, let's say, the environmental agents that we see up on the top. Like we use these every day, but we really need models to assess their safety before we can safely pass it on to the end users. Similarly, the pharmaceutical industry, who's been instrumental in the last few months and before, obviously, they want to understand whether their drugs that they're developing are safe, but more importantly, they need to know, and also we need to know as the human population, whether they work, which is the efficacy part of the whole problem. So I'm gonna try to motivate you in a way, first economically, and this is a graph that I really, really like. This shows on the very right-hand side, we see how the spending in drug development evolved, which was at the 200 million level in the 1980s. But today, and like the last data point is from 2005, but today and that's 800 million. Today, this value is well over $1 billion for a single drug to make it to the market. But if you break it down to preclinical, which is in vitro models, animal models, there's also substantial increases there, but there seems to be more investment, or let's say more work that has, or there's more money spent in the clinical stages. So this made me question whether there's not enough new investment in preclinical strategies, or the preclinical strategies, which is animal and in vitro models, are they not good enough for testing in efficacy, and whether we need new models. And I think that was one of the conclusions that NIH and DARPA made back in the day, in the early 2010s, to build new and better models. But yeah, so basically the gist of the slide is that we're spending lots of money, and it's increasing every year to develop drugs. So then we wanted to look at the source of this spending, like why do drugs fail and how they fail. So this also changed over time. And I'm going to walk you through these one by one. So in the early 90s, the failures for drug development, it mainly stemmed from drug metabolism and pharmacokinetic issues. This is the big slice here at 40%. And efficacy was, even then, really important in clinical failures. A lot of drugs would make it through different stages, but at the end, they were not good enough to make improvements. Back then, the safety and toxicity wasn't as big a deal, but there was a really major shift from the 90s to 2000s, where the safety and toxicity became really important points in clinical failures. And efficacy pretty much stayed the same, but it seems like the drug industry solved the drug metabolism and pharmacokinetic issues. There is then another very important shift, if I can advance myself. Yeah, so in the 2010s, the efficacy has become an even more important problem. And here we're looking at more granular levels, like late stage clinical development. Efficacy is really a huge problem. And in phase 3, which is the very end stage of clinical testing, it is the biggest problem by far. And a similar data, which is from later in the 2010s, is provided by AstraZeneca. But here we can see the problems changing from the preclinical to the clinical stages. In the preclinical stages, most drugs are screened because of their toxicity profiles, which is a good thing. But towards the end in this process, efficacy is always the much bigger problem. And this sort of tells us that, yes, it's good to look at toxicity, but can we do anything about looking at the efficacy of drugs in vitro models or animal models, let's say, early on, so that we don't face these huge hurdles and waste lots of money in the clinical stages where we actually try them on patients, right? So that's the major driver for the research that we conduct in vitro tissue modeling. And then the other reason we choose in vitro is the really disappointing predictive power of animals in toxicity testing, let's say. So we're looking at a graph from a 2000 study by Olsen. And we're looking at whether these different species predict the human condition for drug toxicity. Even if you combine all species to have to predict, the concordance is only at around 70%. The dogs, our beloved friends, they're the best, but even then they only predict 63% correctly. And rodents, which are very, very commonly used in drug tox testing, they predict only with 43% accuracy, which is simply put worse than flipping a coin. And what is even worse is like the mice and rats, who are thought to be pretty similar, they only predict each other with 60% accuracy. So there is obviously an important problem using animals for toxicity screening. Another important problem with animal models is the fact that it's very hard to isolate the individual contribution of cells and their contribution to different mechanisms of action. And these are all reflected in the work in the recent years. Let's say in the European pharma, there's a considerable reduction of animal use, but this is also driven by ethical concerns. And there's an important drive to reduce animals in drug development and also cosmetics, as you might know. And this is outlined in the 3R approach, and it is the reduction, refinement, and replacement of animal models. And so these are all the drivers to develop in vitro tissue technologies to test drugs. I want to answer one more question in this area. And if I can advance. Okay. So the other question that I wanted to answer is, why do we want to use, why do we, okay. Can everybody see, Başak abla?
Yes, we can see your screen.
Okay, I did something, it seems. Okay. So the question that I'm trying to answer here is, why do we want to use microfluidic models? And to answer that, I'm gonna talk about the two parts of this word, which is micro and fluidics. And the traditional alternative to microfluidics is macro, large, and static systems. So macro systems we've been using for a long time. These are your regular tissue culture plates and other similar large models. They really have advanced the field of in vitro tissue engineering. There's no question about that. They're very easy to handle and fabricate, huge pluses. They're compatible with all the imaging and analytic systems in terms of the volume they provide. But they have a very important problem, the macro platforms. And this issue is simply that we use too much volume per cell when we're culturing with these macroscopic platforms. And this reduces all the signals they provide. And it's even more important when you're looking at co-culture systems, the signal attenuation problem, because if they're creating signaling molecules, they're immediately diluted before they can reach the cell that they're affecting. So this is one of the problems that we would like to solve. The other problem, and I'm going to move on to the static system. Yes, changing media every day is easy and it's simple enough, but more often not, it's not quite the right dynamics that they provide, because your body and the tissues in your body certainly do not have one single or simple media change. So this is an important problem in terms of predictive power with static systems. So if we look at the micro side of micro platforms and micro side of things, we think that in the micro platforms, we can reach better media volume to cell ratios because of the reduced height of these systems. This can solve the signal attenuation problem that I was alluding to. But it also creates problems in terms of oxygenation. This is especially important for cells like the liver cells we work with, because they have really high metabolism and they have high oxygen requirements. It's similar for the cardiac or the brain cells with really high metabolism. So that's a problem to solve while trying to solve the other problem. The micro platforms can provide really fine control of the spatial and temporal dynamics in a microfluidic platform, so you can design experiments with better dynamics. And at the end of the day, you use much less volume, much less cells, so they can become cheaper alternatives to the macro platforms. It's also very important when you're using expensive primary cells or the current really expensive iPS-derived cells. So if you look at the flow, the fluidic part of the microfluidic world, we know that it's not very easy to operate them right now. It usually takes a well-trained technician or a postdoctoral level person with technical knowledge, but this is getting much, much, much better in the last 10 years. And we think that on the plus side, as I explained, with the better control of the environment, we can create dynamics that are more similar to what we experience physiologically in the human body. So with that said, I'll go ahead and talk a little bit about what we did in this round. And I will start with the metabolic patterning on a chip, as I would like to call it Mpog. And we're going to discuss how we recapitulate the heterogeneity of liver and its applications in toxicity and drug efficacy, and then disease. So, let me see. Okay, so I'm gonna start again with why we developed this platform where we are patterning metabolic activities on a microfluidic chip. And the driver for this is the spatial and temporal heterogeneity that we see in the liver. Liver is a very rich tissue metabolically, and it's also very rich in its variance across the blood flow axis. And I'm not sure if you can see my cursor, but we're looking at the delineation of zone 1, zone 2, and zone 3, and the blood flow axis here. As we go from zone 1 to zone 3 in the liver, we see wide-ranging changes. It can be gradients in oxygen, nutrients, hormones, and these dynamic effectors can actually change the metabolism of the cells that are in these different zones. Similarly, genetic variance across these zones can affect the metabolism. And this is something we really were interested in, started in 2014-15. And in the healthy tissue, this manifests itself as changes in metabolism, as I explained. But if the changes are in drug metabolic responses, and we know that these drug metabolic response changes across the zones do exist, we know that drugs can have different toxicity based on the change in this metabolism. So this was a huge motivation for us. In disease tissues, and I'm going to give you one example, and that is a non-alcoholic fatty liver disease that's I believe I might have, and many of us in the lab might have. This is a shared problem I think we do have in the lab. But this disease, a non-alcoholic fatty liver disease, which is basically starting with the accumulation of lipids, turns out to be a very heterogeneous disease. As a matter of fact, there's a very recent, and it's heterogeneous not only spatially, as a very recent uh, mass spectrometry study shows, but it's also very heterogeneous in its progression, temporal progression. You start with very low accumulation of lipids, and all of a sudden you end up with really large triglycerides accumulated in your cells, and then it changes all the dynamics. So there is lots of heterogeneity in disease as well. And as a matter of fact, the conclusion of this paper was suggesting that the spatial context is very important in NAFLD. And this was a driver for us to replicate these on a microfluidic chip using the Mpog. So then I want to go and tell you about how Mpog works and what's the principles behind the Mpog. It's actually quite simple. We've taken some old ideas, which is gradient microfluidics. We have a gradient generator where we can create many concentrations, and here it's only 5, many concentrations of the same, let's say, hormones, enzymes, oxygen, and other small molecules. And then we can flow these over the tissue to get different metabolic responses or metabolic capacities in the cells. Our design goal when we were developing Mpog was to have a single tissue. We did not want to look at isolated chambers simply because the liver is not isolated like this. And we wanted to see how the communication between different sections of the tissue can make differences. We also wanted to design such that we could get enough sample for both gene expression and fluidic analysis. And there were like many different ways. We started with a fairly small device where we had very few cells. This was on the order of 300 microns across this channel where we see the gradient. And this 300 microns really was similar to the sinusoid, and it was very simple to image because you only had to image once and you can see a gradient easily. The disadvantage also stemmed from the same thing, that we had very few cells across this device. We could not get enough samples for gene expression, and we could not collect the effluent from this device. So we developed a second version where we addressed these issues, where we created larger channels and distinct but still connected tissue. And that was our version 2, and that's where we're at. The overarching goal for us is to create multi-layer and multi-gradient chips. And this is driven by the idea that we don't only want to have high fidelity, meaning a heterogeneity in the tissue, but we also would like to create many experiments in one single device where we could do we can get hundreds of outputs from a single device. This is very important in the disease that I was mentioning, the non-alcoholic fatty liver disease, because it seems that one of the really important solutions to NAFLD is combination therapies using more than one drug. And in order to test such combinations, we need high throughput combinatorial devices. And our overarching goal is basically driven by this idea. We published quite a few papers in this area, and you can see the references below, but I'm going to go through them in the next 10 minutes. So to answer the question, we're going to go through examples of heterogeneity in normal liver physiology. We're going to look at the zonation of toxicity. Like I was saying, you could have toxicity in one section of the liver, but not the other. We're also going to look at, as I said, the heterogeneity in the disease, NAFLD, and we're going to look at how oxygen could be a factor of lipid accumulation, meaning you can have, just because you have a gradient in oxygen, you can have differences in lipid accumulation across the liver. So I'm just going to walk through those examples one by one. So we're going to first look at the Mpog for normal physiology, where we're going to look at carbohydrates, nitrogen metabolism, and toxicity of a very common and beloved drug, which is paracetamol in Turkey and acetaminophen in North America. So the liver plays a very important role in maintaining the balance of glycogen, which is the carbohydrate stored in the liver, and glucose, and the homeostasis of these two. So it's a very dynamic process. And insulin and glucagon are two of the main drivers of this homeostasis. They're the regulatory hormones, as we call them. And in normal physiology, across the liver, you actually see a gradient of the ratio between glucagon and insulin. So we wanted to replicate this, and we wanted to do an assessment of the changes in carbohydrate metabolism using a PAS staining, periodic acid shift staining, which actually shows you the glycogen storage in the liver. So we actually established this using both primary red cells, hepatocytes, and primary human hepatocytes in the chip that I have shown. Here we have a gradient from 100 nanomolar glucagon on the left with 0 insulin, and on the other side we have 0 glucagon and 100 active per liter insulin. And these are two extremes that we wanted to work with. And the results, I think, speak for themselves. We can see that we have significant changes in glycogen storage across these two extremes. More so, perhaps, it's clearer, but when normalized, I think the results are pretty similar for the red hepatocytes than the human hepatocytes. So the response is not exactly the same unless they're normalized. So we were able to replicate the zonal-like carbohydrate metabolism simply by replicating the hormonal gradients in the liver. So this was a good demonstration that this chip was useful in recapitulating some parts of liver physiology. We then moved on to nitrogen metabolism. The nitrogen metabolism, as I'm showing here, is the conversion of glutamine and ammonia through the ornithine cycle to urea. And once again, the metabolism of nitrogen is driven primarily by the ratio between glucagon and insulin levels. And so we did something very similar here. We have the same gradient, but this time we assessed the metabolic changes by a pseudomarker, which is an enzyme in the ornithine cycle. And once again, we see considerable changes across the device, both in primary red hepatocytes and the human hepatocytes. And you can see more about this in Abraham's work from 2018. So one of the more important probably things for the drug industry, because that was our motivation before, is toxicity of drugs. Usually when people do tox tests, they only replicate one zone of the liver, and this usually is zone 3, sometimes zone 1, depends on how they're doing things. But the gist of this is that liver toxicity is not homogeneous. It varies across the blood flow axis. And basically, the example we chose to demonstrate this is paracetamol. And the way the toxicity for this drug works is that it's metabolized through the phase 1 drug metabolism. And then its intermediate product, which is NAPQI, can be toxic to the cells. So we wanted to replicate a dynamic system like this. And we use the well-known inducer of CYP1A1. And this is 3MC. So we use 3MC on one side and a high dose of 3MC on the other side. This is from our previous, like a smaller device. And we created difference or a gradient in the activity of these enzymes. And surely enough, we were able to see, like, we're applying the same acetaminophen exposure to all the cells across this variable drug metabolism tissue. And we can see a distinct difference in the toxicity in the zone 3 cells, which is what's expected. The higher the CYP activity, the more intermediate products you produce, which in turn kills the cells. So we were able to replicate this, and this is coming from Bill's work from 2016. So this was another good example, demonstration for us that we can actually capture some of these normal physiologies and toxicity response. So I'm gonna now move on to disease models. And the disease that we have chosen, which is really interesting to us, is non-alcoholic fatty liver, and the more progressed form, which is accompanied by inflammation, is non-alcoholic steatohepatitis. As I've introduced before, this is a very complex, progressive, and heterogeneous disease, and its prevalence is through the roof. Nearly 25% of everyone in the world has non-alcoholic fatty liver disease. This is the first observation, about 5% of your cells being fatty, that meaning accumulating lipids, triglycerides. But I think the more concerning part is when the disease progresses, which is accompanied by inflammation, and which in turn triggers your macrophages, the Kupffer cells in the liver. And then there's a whole cascade which becomes very injurious to the tissues, which then progresses to cirrhosis, and you'll eventually have cancer, you'll need a liver transplant. So basically the downstream of this disease, unless you start taking care of yourself or there is some intervention, is quite fatal, actually. But so, like, we wanted to understand this very heterogeneous disease, and like the problem is that because it's so complex, the disease, we thought that we can start modeling it in simple terms and ask simple questions. So that's how we started. And let me see. Okay. So this was a first attempt using the Mpog device to create, like, we wanted to recapitulate the temporal or the spatial heterogeneity of non-alcoholic fatty liver disease. And like I said, we wanted to ask very simple questions. So the question that we're trying to pose here is, can we create on a single tissue variable lipid accumulation? And we wanted to do this simply by introducing different levels of free fatty acids, which could be in our diet? And can we induce different levels of lipid accumulation on a single tissue? We then wanted to assess whether there would be, like, we could recapitulate corresponding metabolic and gene expression changes in the cells. So this was basically a first demo that creates heterogeneity of the disease in a single tissue. Then, like I said, oxygen could be an important modulator of lipid accumulation because, frankly, the lipid accumulation is a balance between the fatty acids oxidation and the de novo generation of lipids. So we wanted to see if we can create in vivo-like responses through the oxygen. So this is our first attempt. And as you can see, here we're using a 24-hour application of linoleic acid gradient and the subsequent staining using oil red O, which stains for the lipids in the cells. On the top, you see device where we apply no gradients and there is no linoleic acid. On the second row, we have the gradient. And on the third row, we're looking at a phase contrast image. As you can see, we can indeed create on a single tissue. This is coming from a combined single tissue. On a single tissue, we can see variable lipid accumulation from left to right, just responding to the excessive free fatty acid administration. This would corresponding to me eating lots of French fries, burgers from McDonald's. And if we look at the inset here, we can actually see these even with the naked eye, the lipid accumulation. And just to confirm, we did, we quantified this accumulation in the chambers, and we also made sure that our cells didn't die because of this excessive accumulation of lipids. Then, like I said, we wanted to look at different genes, and these genes, some of them are quite novel targets of target for therapy. And I'm not going to go through in detail in all of those. And we actually picked these up based on their growing importance in the field. The first set is the genes that's related to the generation of lipids. And what we see, for example, for this ACACA is that it directly responded within 24 hours to the gradients that we applied, and the expression in the gene change such that more fatty acids would be synthesized, meaning we would have more lipogenesis actually. And that's replicated in the picture that we show where we had more lipids. A similar story is for the FASN. These two are very novel targets for the drug industry. And we hope that one day we could use this platform to test the efficacy of drugs. Basically, we would create this gradient and then we would try to normalize it or reverse the effects of the fattening by the drugs that they want to test. Similarly, we had two more. One of them is a master transcriptional regulator of the lipogenesis, de novo. And the response here is different. It's not very linear. This one seems to be an on and off. So not all the responses are linear. And this is probably because of the transcriptional factors getting regulated, which in turn regulates these genes. But if we look at the GCK, we see that that is more responsive. also by the amplitude. But all of these show that there is more lipogenesis going on when we administer more free fatty acids. We then looked at the fatty acid metabolism related genes. We see a similar story here. And these actually eventually can, like some of these also affect the downstream diabetes, and they can be upregulated in carcinoma, as I've shown in the progression of the disease. We also looked at the changes in transporters. These are both the fatty acid transporters, but more importantly, drug transporters. The drug transporters are important because they regulate how the drugs get in and out of the cells. So this is an area where we're trying to grow this work, but that'll be part of the future as well. Once again, our goal here is to eventually make a platform where we can look at the efficacy of drugs. Another important question, like I said, is like across the liver, we see a very significant gradient of oxygen. It goes probably from around 9 to 4% oxygen saturation. And it's not the 20% that most people will use in their classic experiments. So this is actually quite important. Oxygen, the availability of oxygen really changes the dynamics. And that's what we wanted to show here. The idea is that if you have more oxygen available, which is the left-hand side in our device, as well as this cartoon, you'll have more fatty acid oxidation. Therefore, you won't have as much accumulation. And this is actually quite straightforward, but and on the right-hand side, the zone 3 part of the liver, you have, because of the consumption by the cells, you'll have less oxygen available, and therefore the zone 3 hepatocytes are expected to be more fatty. So we actually used a very direct approach to creating oxygen gradients. This is in contrast with many people using nitrogen, excess nitrogen. We use chemical quenchers, and we created a gradient of these quenchers. This is a sulfide cobalt mixture. And we created a gradient of oxygen that's in the range of 11 to 7. This is somewhat close to the physiological ranges that I was mentioning. And then we superimposed on top of this fixed levels of linoleic acid. Unlike the previous time, this time we have a gradient in oxygen, but we superimposed a steady level of linoleic acids. And for each of these, we actually did see the oxygen to have an important effect on the lipid accumulation. However, the highest effect was actually at the lowest concentrations of lipid accumulation, which actually also corresponds more to the physiological level, 0.25 to 0.5. But 1 millimolar could correspond to a severe level of free fatty acids availability. But basically, we actually recapitulated responses that are very similar to in vivo work and also computational work people have been doing. So this was really a huge plus, and it was really welcome by me and the people who did the work. Beza was the lead on this. So where are we going with this? As I was showing, and also in the NAFLD realm, as I was showing, our goal is to create higher throughput models, and that's something we're doing. But in the NAFLD realm, we're also trying to translate this to humans, we've been working with rats for the NAFLD. And recently, we have been working on creating human models where we really dissect the inflammation response as well as the drug enzymatic expressions in these cells. And moreover, we have been trying to understand how the drug-induced enzyme changes are regulated in NAFLD, which is really important when people are taking more than 2 drugs, because the effect of one drug can diminish the effect of other or create toxicities. So this is where we're going in this realm. So I'm going to switch gears now, but I wanted to thank all the people who worked on this trust. I'm going to move on to biomaterials, but I'm not sure how I'm doing on time, and I think eventually I should get to the deep supercooling. Başak abla, what is the time that I have left?
You have about 6 minutes left.
6 minutes?
Yeah, you can. No.
Okay, so this is actually really interesting work, but I think I'll have to skip over it. This is where we looked at how we can solve the problems of PDMS, and this is the work of Aslan Gökaltun. PDMS is super problematic when it's used for with proteins and the testing of small hydrophobic molecules, which are drugs. And we wanted to solve this problem, and we used a very novel approach where we blend in copolymers into the PDMS. And basically, we created a solution which is biocompatible. It reduced the wetting of PDMS a lot and had a shelf life of 20 months. And it also reduced the protein adsorption by a lot. You can find all about this work in Aslan's work from 2019. And she's improving this now with Zwitter-ionic polymers. And this work was actually with my collaborator, Dr. Ayşe Asatekin from Tufts. And frankly, the lead here was Aslan, who's still with us and pushing this idea forward, making PDMS better. Okay, I'm gonna switch to biopreservation with my 4 minutes left. So I want to talk a little bit about supercooling today. Basically, supercooling is an intermediate solution between cold storage, which is very limited in time, and cryopreservation, which promises infinite time, but usually falls short for some cell types and also for whole organs. Supercooling is a metastable, or supercooled states are metastable states for liquids below their melting points. And it can provide an intermediate solution where it will be longer than cold storage, but it will be high quality, and it could really work on organs too. As a matter of fact, Dr. Uygun, Korkut Uygun, and colleagues, they showed in 2014, and I was a very small part of this work when I started with Dr. Uygun, show that they can actually extend the preservation time of livers by keeping them at -6 degrees without any freezing. This is quite a feat. Not only that, they were also able to transplant this after 3 days with 100% success. This is more than tripling the preservation time. I did a lot of work in this realm on the cells of the liver. I'm not going to go through this because, frankly, the exciting stuff came later for us. Dr. Uygun expanded this work to human livers recently, which was amazing. It's up to 27 hours for a human liver now, which is, again, tripling it for the human liver. And even the NIH director talks about this. I want to talk about deep supercooling, if you allow me, Başak abla, for a few minutes.
Yeah, please continue.
Okay. So the deep supercooling, I'll start with, again, by the bottleneck in supercooling. Uh, we can easily reach -4 to -6 degrees by the current preservation solutions that we have, which is UWHTS. But reaching lower temperatures have been very hard. Reaching lower temperatures, like the homogeneous freezing point of water at -40, it's only possible for tiny volumes of droplets. But this also limits the applications in cells and organs, because we cannot reach temperatures below -6 easily for long times and large volumes. So this is basically the problem that we had when we started the deep supercooling work. What we wanted to do was to extend the temperature range to below -10 in a stable manner. Similarly, we wanted to expand the volume, and probably also very importantly, we wanted to go from like few days, hours, to weeks and months. So this could be a really good potential alternative if we can get to those low temperatures for cells like red blood cells, and also some exotic cell types which are really hard to cryopreserve. What we did initially was and so I'll actually go through the experiment so that it's easier in the limited time we have. Here you see a series of samples where we had water without anything on top of it. Then we started putting different oils on top of the water, and we kept all of these, and these are 1 milliliter samples, at -13 degrees for up to 7 days. And the cumulative freezing frequency, basically the events, freezing events for these samples, was near zero for all of these samples that are sealed by oils, except the olive oil, which curdles and sort of freezes itself. But even that had a suppressing effect. So our hypothesis was that probably nucleation of ice is much higher at the double, triple interfaces where air, solids, and water meet. We also think that sealing aqueous solutions with these liquids, oils, has an additional benefit of dissipating mechanical and other types of disturbances at the interface, which is very prone to nucleation. So this was a recent publication in 2018, and that's the work of Dr. Heishui Huang. We showed that the volume dependence of this method, the oil sealing, is very, very low, which is sort of counter to the classical theories. Up to 10 milliliters, the volume dependence was extremely low. And if we use the paraffin oil rather than the mineral oil, up to 100 milliliter at -13 degrees, we had very low volume dependence. But at the lower temperatures, the volume dependence was stronger. So I'm going to skip over this. We also showed that there is a critical sealing. This was to show that it's really coming from the interface, this effect. So we added increasing surface coverage of oil on the aqueous solution below. There is a transition, and this gray actually corresponds to this figure here, this picture, where we have a very little bit of water exposed to air. And even though the nucleation has decreased, it's not fully decreased. And we're doing a temperature sweep here, and we can actually suppress nucleation if we cover these samples fully down to -16 and -20 with low nucleation probability. So this was really an effect at the interface. This is what we wanted to show. We pushed this even further, and we were, so oils are basically mixtures of alkenes and alcohols. So what we did is to look at pure hydrocarbon sealing of the water samples. With alcohols, we showed that with small chains, short chains of alcohols, we can have this effect, like very strong effect, even at minus 20 degrees for a week without any freezing. With the alkenes, we showed a different dependence on chain length, where the longer alkane chains were much better at suppressing the ice nucleation. You can definitely find more about this. We showed that the oils were very stable. Like I was saying, they dissipate disturbances such as centrifugation, just dumping the samples into 37 degrees directly from minus 20. But we showed that the alcohols provide the utmost stability, where even if you ultrasonicate, these are really high frequencies, over 20 kilohertz, they still do not freeze, even if you do these torture tests to them. So this was really quite exciting. Then we looked at some applications of deep supercooling, where we kept mesenchymal, adipose-derived stem cells at low temperatures. We showed some success here, where the deep supercooled cells did better than the cold stored cells with good attachment. The lower the temperature, the better cell culture attachment. And you can see more about this in the paper cited below. More importantly, I think we set out to extend the preservation time of red blood cells. Currently, they're only preserved for 42 days. But we wanted to do better and alleviate ethical concerns. I say ethical concerns because, frankly, the quality of cold stored red blood cells after 28 days is a question mark for the clinicians. So we had some preliminary success here, where we were able to supercool red blood cells up to 100 days. This is more than double the 42 days at minus 16 to minus 13 degrees. So this is more of the success, showing the same success. Basically, it seems like we can extend the preservation time of red blood cells with really good quality. And this is something we're working on right now. These are just some conclusions. Supercooling is really a good alternative for organs, as Dr. Uygun showed, and also some of the more exotic cell types. We're currently working on many different applications of the deep supercooling. And I would like to thank all the members that worked on these projects. Heishri was the one that started it. And today we have new people like Aslan, Delaney, and Zia carrying on the torch. They're working on advancing this in the lab right now. With that, I'm going to thank you, and I apologize about going a little over time and accelerating in the end. I want to thank all the people listed here, and especially Başak abla.
Thank you. Thank you, Berk. So that was a great talk covering a lot of different research topics. So there is one question in the chat from Justina. She's asking, how many conditions can you currently test on Mpog? I think you showed some evidence later on in your presentation that you can do more deeper.
So the prototype, thank you, Başak abla. So the question is how many conditions we can test. Currently we can do 5, but the idea was to go to 8, as I've shown, and then do it 8 by 8, meaning we'd be able to do 64. But frankly, it's all it needs is some more engineering to make it robust and make it more high throughput.
Mm-hmm. And you can collect the cells from different parts of the channel.
Yes, we have done that. Like we've done pretty primitive approaches of dissecting them after like removing.
Mm-hmm.
But we have experimented with lysing them by putting separators. So yes, we can collect them and then do gene expression analysis.
Mm-hmm. Okay, so we can take a few more questions. So if you have any questions, please type it in the Q&A. So I was just wondering, in the NAFLD model, I mean, it's good that you are developing this platform for drug testing, but I was wondering if you can test, you know, lifestyle changes, how it affects the liver cell morphology and the lipid metabolism. Because, you know, a lot of clinical studies show that if you change your diet and, you know, do more exercise, make sure you get enough sleep, they all help resolve the fatty liver. So can you share your thoughts about that?
Sure, I can share my thoughts. I think like where we are right now, so the experiments were limited to a day or two. So just based on that, no, because the lifestyle changes would take months, years to take effect. And frankly, doing those in vitro is hard, but I mean, they can be simulated, but then we need to extend the model to cover like at least a month, I think, so that we can test these. I'm not sure how we do exercise. Probably we could change oxygen levels, like simulating exercise. That is a possibility, I think, but at this point we cannot. But it's really interesting that you brought it up.
Yeah,
I think we should think about it and do it.
Yeah, I think you should think about it.
Yeah. I mean, the
Because if
dietary
you can
ones are
find
easier.
something that, you know, mimics what exercise does, you know, with a drug candidate, then that would be a good option for lazy people. Okay, so there is one question.
Yeah, I see. I think Urjan has asked a question. Yeah. The question is, what are the main challenges transforming Mpog to a high throughput tool for testing? Like I was saying, it's mostly engineering problems. I mean, this was a prototype, and like I said, we used very primitive methods to show proof of concept. Like the fluid collection is not always very easy. Abraham experimented with many different methods, leaving it out, collecting it with a pump. So yeah, there are lots of kinks to be worked out in terms of sample collection. The other thing is, like, we really need to model these systems well in terms of the diffusion of, let's say, the oxygen, the nutrients, or the inducers that we're using, so that we actually are getting the conditions that we think we are getting. And this is something we did with the smaller model. But for the larger models, this needs to be confirmed and reconfirmed, because if the cells are slightly different, the consumption will be different. So these are all the things that we can do. But it needs a lot of fluidic engineering. We need good engineers like you to improve the platform. And I think we just got one. So I'm very happy about that.
All right. So I don't see any more questions from our audience, and for the interest of time, I don't want to keep you and the attendees longer. So I just wanted to thank you again, Berk, for joining us.
My pleasure.
And sharing your work with us. And I'd like to thank our audience for their attention and participation in the discussions. As I said at the beginning, this is the last talk of the semester in this seminar series. We're hoping to be back in early March, and we'll be sharing information about the speaker lineup ahead of that date. So thank you again, and it was nice seeing you.
Likewise. Take care, everyone.
Bye-bye. Bye-bye.